Okay, so today I'm going to show you what you need to know for your nervous tissue histology lab, and then I will give you directions on, and the directions are in the lab. You'll figure it out. So what we're going to do is we're going to go through the histology parts. I'm going to describe to you what you're going to see, and then I will actually show you the slide that you will be labeling. But I will not give you the answers because that's not my jam. Okay, so the first thing we're looking at is an ox spinal cord smear, which is like literally what it sounds like. There's a little piece of bovine spinal cord, which is put on a slide, and another glass slide is sort of slid over it, and that literally smears it out. One of the things you get to see in this is that you separate the cell bodies from each other. Remember, we talked about how they stick together in the central nervous system in clumps called nuclei in the central nervous system. And so when you separate them out, then you can see them individually. So you see here you have, and so this is what you would see typically in the central nervous system. You have a cell body, which is the main part. This one says that this is labeling this as an axon. When you smear it, you really sort of lose the identification of dendrites and axons. So I would ask you to call all of, unless it's someone tells you the answer, I would say that all of the processes that stick away from them at the cell body are all called cell processes, because you just really can't tell, or neuronal processes, which one's a dendrite and which one's an axon. Within the cell body, there is a nucleus, which is sort of this light haze here, and a dark spot in the middle, which is the nucleolus. That's frequently called an owl eye, just because it has, sort of looks like an owl eye upon presentation. There is sort of this blotchiness within the cell body. Those are Nissl bodies. Nissl bodies are just a fancy name for the endoplasmic reticulum of a neuron. Outside, you see glial cells, probably astrocytes, kicking around, waiting to, support this cell body. All right, so that's pretty much what you're looking to identify. You will get this one. So again, you're looking for the cell body, which would be the main body. I can't think of a better word for that, substance of the cell. Inside of that are the Nissl bodies, which are the little splotchiness. There are glial cells on the outside, which you can see their nuclei of. All of the things sticking out are the cell processes. Inside the cell body is the nucleus, and inside the nucleus is the nucleolus. So you'll label all those, and there's a place to do that on your lab. And this is all from activity 1. So if you took one of these cell processes and decided it was an axon, and you went out to a myelinative nerve fiber, there's always this osmium stain. This is like very typical for nerve fibers, just presents nicely, but it also presents in black and white. And what this is a nerve. So say your sciatic nerve in your that goes from your hip down your leg, and it's sort of like a block. It's like a bunch of different axons together, and so they go and they take two pins and sort of tease them away from each other so you can see them individually on a slide. So that's how this is created. And what you're looking at is a whole mess of axons. They're all running left to right here. And I always think that they look like sausages or hot dogs that are like strung together like they are in cartoons, and sometimes at a deli, I suppose. And so the places where the whole hot dog is like a Schwann cell, and within that Schwann cell is the axon, which is darker. You can really see it in this one. And in between that, where the Schwann cell goes away, or the myelin goes away, is the node of Ranvier. Collectively, if you sort of made a bracket on the outside of all of this, that would be the axon. That would be the myelin sheath, and each of the individual hot dogs is a Schwann cell. So This is not the one you're going to label in your lab. There is one that looks like this, where you are looking for the axon, which runs through the middle of all of them. The node of Ranvier, which is not too subtle in this picture, where Schwann cells end and the axon is there just by itself. Collectively, the whole of the outside would be the myelin sheath and a Schwann cell. And here you can see the nuclei of the Schwann cells. They are darker within that Schwann cell sheath. All right, so there are your myelinated nerve fibers. So that's activity 1, so looking at a typical neuron. Now we're going to look at specialized neurons that you see in different parts of the central nervous system. And the first one is a Purkinje cell. Purkinje cells are cells in the cerebellum, which is at the base of your brain, and they are cells that connect in the cerebellum. The cerebellum is part of your brain that plans motor coordination. So if you have problems with your cerebellum, you lose coordination and motor function. So there are layers to it, and the outside layer is here. So this is like the outside of the cerebellum, and the outside layer is called the molecular layer, and it has way fewer, it's less cellular. You see that there are many fewer nuclei there. And then this more deep to that is the granular layer. And the granular layer has lots of nuclei, and it's very sort of dense looking. And then this is all on the cortex, which is the outside of the cerebellum. And then between those two layers, the molecular layer and the granular layer, is your Purkinje cells. And they are always oriented with the cell body on the granular cell layer side, with the dendrites reaching up into the molecular layer. They branch a lot. The next slide you'll see a lot of that branching, but they're super branchy. You'll see in the slide that you label, and they form that border, and they're really important in coordinating motor movement. So this is one of my favorite histology slides ever. It is much different, but it's oriented the same. It's much different in its staining, but it's oriented the same way. So you have that granular layer, which is very cellular. I will tell you that the nuclei in this are sort of a hot pink fuchsia color. There's a molecular layer, which has many fewer nuclei. And then you have Purkinje cell bodies in between, with the dendrites reaching into the molecular layer. And where the dendrites spread out is where you have dendritic spines. So like the ends of them are the dendritic spines. And this is a really nice slide because you can see the branching and how branchy it is compared to the last one where it's really subtle. All right, so that's what you'll label in your lab. Your next slide is pyramidal cells, and pyramidal cells are called pyramidal cells because the cell body, which is this part here, is very kind of pyramid shaped. And I will tell you, once you see the pyramidal cells, you'll hopefully recognize them again. And when they talk about, like, you know, you watch TV or you watch like a commercial or something and they're like, cells in your brain, for some reason they always pick pyramidal cells for that. And they use this stain that is sort of looks kind of sepia toned if you're into photography. So it has this kind of yellowish tone to it. So there is a cell body with an axon, and the axon kind of comes out of the middle of the cell body. And the next slide will be the one that you label will be oriented in the same way, pointing down. And then there is an apical dendrite. Apical always just means primary. So it's the one that comes out of or is, you know, I guess in directional term is leading directly to the cell body. And then Where it branches from there, it would be the basal dendrites branching off of the apical dendrite. And then if you can see the ends, those are the dendritic spines. These always look like trees to me. And these are in the cerebrum, which is the main part of your brain. And it helps vision-guided motor function. So if you anything that you need to do where you were looking to see what was happening, to guide your motor movements. So you're saying, I'm going to walk across a cluttered room. That's definitely a vision-guided motor function because you need to be able to see where you're going so as not to trip and fall. So that's what pyramidal cells do. So this is the one you're going to label. The cell bodies, again, are sort of the triangular cells. Pick one, the most pointing down-ish cell process off of that, and that is the axon. So pick one with a good pointing down, and they're oriented the same way as the last slide. The apical dendrite comes directly out, and if you look carefully, there's one or two that branch. I don't think that you can actually see dendritic spines on this, so I think that you can do without labeling the dendritic spines. All right. Again, that's activity 2, pyramidal cells. So the third slide is dorsal root ganglia. Dorsal root ganglia are a bunch of cell bodies collected in the peripheral nervous system. Generally speaking, they are pretty close to, there's a lot of them clustered near the spinal cord, and they look much like this. So you have a cell body, you have actually a bunch of cell bodies here with a nucleus and a nucleolus. You have around the outside of them satellite cells. Remember those from before. Remember that they're part of your peripheral nervous system glial cells. And then this stuff that looks wispy are the axons. So the axons of other cell bodies that are running by these cells. So these are the cell bodies, and then you have axons collectively from other ones, from other cell bodies that are in other parts of this ganglia. So, and the dorsal root ganglion brings sensory information from receptors in the peripheral nervous system into the central nervous system. So, again, cells, cell bodies, nuclei. In the middle, you have nucleoluses, cytoplasm between clearly the outside of the cell and the nucleus, rings of satellite cells around them, and then axons are sort of the wispy things. So you have axons up here too. All right, so again, this one I think the hardest thing to find in this picture are the axons. Look for the wispy things. You're looking for the big circles being the cell bodies, littler circles inside that being the nucleus, the tiny circles in the very middle being the nucleoli, cytoplasm between the nucleus and the outside of the cell membrane, halos of satellite cells around the outside, and then look for the wispiness for axons. And those are your dorsal root ganglia. All right, so if you took a peripheral nerve and you did a cross-section of it, that's what we're looking at in activity 3. If this looks like muscle to you, good job. It should look like muscle to you. And they have the same connective, we haven't talked a lot about this, but it will be on your test, not your midterm, but your test on the nervous system. There is connective tissue around the outside of a nerve. So this is a cross-section of a nerve. Around the outside of the whole, this whole thing is a nerve. Around the outside is epineurium. And then there are fascicles, just like you had muscle fascicles, except for it's a bundles of axons together. And those bundles of axons are called fascicles. And then on the outside of them are perineurium. So fascicles here, here, and here, and around them are perineurium. And then if you zoom super close in on that, you can find the axons, and the only thing that's different about them is that they have this halo around them, which is the myelin. The endoneurium is on the outside of the myelin sheath, so you have a connective tissue on the outside of that. So you have endoneurium around the myelin sheath, you have axons, you have a bunch of axons together with their myelin sheaths, and Schwann cells in fascicles. Around the fascicles is perineurium, a bunch of fascicles together is a nerve, and on the outside is epineurium. So we're first going to label a low magnification, so you can find one whole nerve. There are multi-sized fascicles on here. Around each fascicle is a perineurium, and around the nerve itself is an epineurium. So this is your low magnification diagram, or histology. And then there's a much higher magnification. Here you can see an axon, which will be in the middle of myelin. The myelin is never going to pick up stain, so it's always going to be a lighter halo around the outside. Around the outside of the myelin is the endoneurium. And then if you look for a axon that does not have a Schwann cell around it, so it's just by itself, there might be an arrow pointing to one, that would be a node of Ranvier. Alright, so that's all that you have to label. Make sure you answer the questions. It's described pretty clearly in the lab. And if you have questions, you can always ask.